density.
Yeah.
1920 density. And last year we had Shukanto Das, also the same batch, give a talk. And today Shomeek is here, he's from Virginia Government University. Last year we had Shukanto Das. Apart from his work, we'll also talk to you about how to get to US and about applications and all. Because as a teacher, I have been giving recos during this time. Per day, it is today just given reco for Sunday and last year. At least 3 to 4 recos per day. I have still not been able to figure out what makes some people click and some people not. It need not be a student, need not be a gold medalist, and yet they have made the right moves and got into the best universities for PhD. I have not figured that out, but hopefully because Shomeek is there in the land of plenty, he will be able to tell us. Okay, with these few words, I welcome Shomeek. Thank you so much for coming.
Pointer. Meat. Pointer. Pointer. Meat. Hello. Hello. Yeah. First of all, thank you, Kushana, for the introduction. Hello, everyone. Hi, I'm Shomeek. I'm currently a PhD student at Virginia Commonwealth University, situated in the city of Richmond in USA. I'm part of the doctoral program which is called Integrative Life Sciences. And those of you who don't know, I guess Pushpaya already said, so I was part of the first batch, 2012 to 2017. So I remember, so basically I was a student, I used to sit on those chairs at some point of time, people used to come here and give lecture. So I'm a bit nostalgic, but as well as happy and excited. So my PhD supervisors are Dr. Peter Utz and Dr. Stephen Fong. I'm working on this problem, which is called Do Protein-Enzyme Interactions Affect Metabolism? It's a computational biology study, and in CMB, we get the opportunity to do research training apart from your parents' institute. So from the first year, when we joined back in 2013, I went to a research training lab where I basically got exposed to the field of computational biology, and I like the field. And that since then, I've been just continuing in that field. So that's the problem. Do protein-enzyme interactions affect metabolism? First, I would like to start by giving some basics, theoretical aspect about my project. So I'm working on one side. Yeah. Hello. Hello. Hello.
Can you change the mic, please?
Yeah, everything is fine. Hello. Sorry for that. That's right. All right. So first, I would like to go through some of the theoretical aspects about my project. So protein-protein interaction, as the name suggests, it's basically one or two proteins. They like to interact. They can interact using electrostatic forces, hydrogen bonding, hydrophobic. So basically, whenever there are proteins, they like to interact and they make protein-protein interaction. And when many proteins interact, they ultimately make protein-protein interaction network. As you can see, one picture on the right. Protein-protein interaction network, they take various forms. You can see them in electron transport, where electrons are being transported through the proteins, which occurs electron transport chain, which occurs in the membrane. You can also see that in signal transduction, excess ligand binding to the receptor, signal propagating to the proteins, transcription factor getting activated. So there also you see protein-protein interaction, and of course metabolism, which is my work, which I will be explaining. Protein-protein interaction, generally there are two types. One is binary interaction, where just one protein interacts with other protein. So that's like binary. It's two, one-to-one protein-protein interaction. And also complex interaction, that is more than two proteins, 2, 3, 4, or whatever number. They interact and they make some complex. So multi-protein complexes. So interaction can happen in two ways. This is just a picture from a paper where I put this here to basically mention the significance of protein-protein interaction. So the boxes in the red, these are the enzymes or metabolic enzymes of methionine cycle, and they are basically interacting with different kinds of proteins of different function. So the boxes in the blue are the other proteins. And this paper basically showed that the enzymes which are in red, metabolic enzymes of the methionine cycle, they like to interact with different kinds of protein and help in their functioning. For example, MAP alpha 2, it's an enzyme of methionine cycle. It interacts directly with BCL2, which is an apoptosis protein, and helps in apoptosis. So protein enzyme interaction, it can play a crucial role in very different kind of processes. So going forward, the other side is metabolism. When we talk about metabolism, it's basically we take anything, we take food, it gets digested in our body, it gets broken down, and then energy is produced, ATP is released, which helps us to do all kinds of life activities. So that's the biochemical process, that's the basic definition, metabolism. If you dig a bit deeper into metabolism, they are nothing but they are just substrate to product reaction, enzymatic reactions governed by proteins called enzymes, or we can say metabolic enzymes. So metabolite 1 converting to metabolite 2, 2 to 3, and that's why it makes a chain of metabolic reaction or enzymatic reaction, and each of the reactions are always governed by a metabolic enzyme. And when these chain of reactions, they form a big system, they can be organized into metabolic pathways, which are nothing but a chain or series of metabolic reaction forming into pathways. And KEGG, it's a database, Keto Encyclopedia of Genes and Genomes, which I use a lot because they have classified this metabolome. Metabolome is basically all the metabolic system in your body, we call that metabolome. They have classified the metabolome beautifully. And the third point takes that definition from KEGG, where they divide the whole metabolome into two types of pathways. One is general pathways like carbohydrate metabolism, lipid metabolism, amino acid metabolism, and then each of these broad pathways can be divided into smaller pathways like PCA cycle, glycolysis, pyruvate metabolism. They come under carbohydrate metabolism. Lipid metabolism, fatty acid synthesis, fatty acid degradation. Amino acid metabolism, aspartate and threonine metabolism. So there are big pathways, and in each of these big pathways there are some smaller pathways. So that's the idea of metabolism. So going forward with my work, so as I said, you can just see it's a substrate to product reaction. Glucose getting converted to glucose 6-phosphate. Glucose is a substrate, glucose 6-phosphate product governed by the enzyme, which is actually hexokinase. So it's shown by a green box enzyme. This enzyme is a protein which is also interacting with other proteins, which you can see protein 1, protein 2, protein 3. So if you just think in a comprehensive scale, that's a computational biology or systems biology idea, like doing data analysis, visualizing everything on a broad scale. I'm basically exactly focusing on the interface or middle of two kinds of networks: protein-protein interaction network and metabolic pathways. Protein-protein interaction network, these proteins which come from the protein-protein interaction network, which are binding to a certain kind of proteins, enzymes, which are metabolic enzymes, and these enzymes are governing the metabolic reactions, which ultimately make the metabolic pathways. So this work which I am doing, it's basically also it will help us to decipher some new mechanisms where we try to understand the crosstalk or the connection between two globally known biological networks. protein-protein interaction networks, and metabolic pathways, where metabolic enzyme is the common link. So that's the idea, and this is from the keg. So when you just if you go to the keg and if you want to see the metabolo, so this is the whole biochemical cycle which happens inside one cell, and that's how metabolism looks like inside one cell. So it's quite big, and so this is basically the carbohydrate metabolism. You have lipid metabolism, something around here, amino acid metabolism, and yeah. So now, since I'm talking about metabolism, the important thing which comes to my mind is regulation. And it always happens basically in order to maintain a constant supply of energy, in order to maintain a constant supply of blood glucose, homeostasis. Metabolic regulation happens where the synthesis of product or the degradation of product, the rate of the reaction is being altered, and we see that in metabolic regulation. And as I said, the governing factors of the metabolic reaction are always enzymes. So any regulation which happens, it has to happen through the metabolic enzymes. So, and the most studied biological process when we talk about metabolic regulation is allosterity. It's basically another allostatic factor, which can be a protein, which can be a chemical, which can be a metabolite. They go and interact with the metabolic enzyme, and then they change the enzyme activity, and that's why they regulate metabolism in that way. So that's the most studied phenomenon. But when I was working on metabolic regulation, I came to know that metabolic regulation can also happen through localization, but that is by transferring the metabolic enzyme at a certain point to do a certain reaction. Gene expression, that is by regulating the gene of the metabolic enzyme, regulating the expression of the gene, which basically codes for the metabolic enzyme. Post-functional modification. And when I basically worked on more, I found that each of this regulatory mechanism, allostery, localization, PTM, gene expression, can also happen through protein-enzyme interaction. So I found these papers where people are discussing how protein-enzyme interactions can play regulatory roles through each of these processes. I will just explain the first one, like trigger enzymes, bifunctional proteins. So what they do is there is one gene called PTSG, which is a metabolic enzyme. It converts glucose to glucose 6-phosphate. Now what happens is when there is low glucose levels, we doesn't want the glucose to be processed. So PTSG has to be inhibited. So what it does when there is low glucose level, PTSG gets phosphodilated and it gets inactivated. PTSG or at the same time also interact with a transcriptional repressor called MLC. So when it binds to MLC, it binds and sequesters the MLC, and MLC can't do its function. But when there is low glucose levels, because glucose is being converted to glucose 6-phosphate, low glucose levels, we don't want glucose to be processed, so PTSG has to be inhibited. PTSG gets phosphodilated, it gets inactivated. When it gets inactivated, it can't bind MLC. Now MLC is a free transcriptional repressor. It goes and sits in the DNA and inhibits the expression of PTSG. So that's how gene expression regulation is happening through protein-enzyme interaction. These are the two papers where also people have explored. So again, in order to assess the importance of protein-enzyme interaction, we basically started this work. But also, like these are protein-enzyme interaction, it all looks good. But is it enough to basically just go and find protein-enzyme interaction regulating metabolism? Is this sufficient reference? And that's answer is no. So there were some previous works. Peter Roots is my supervisor. He's part of all these 3 papers. They did some experimental enzyme assay-based experiments, and you can just read the title and understand what they are trying to say. Is the first paper, the phosphocarrier protein HPR of the bacterial phosphotransfer system globally regulates energy metabolism by directly interacting with multiple enzymes in Escherichia coli. Similarly, the urinaryl transferase GLND and tRNA modification GTPase MNME, these are enzymes allosterically control Escherichia coli, polyglutamic synthesis for C. So these were small scale experimental enzyme acid based experiments where he showed that proteins bind to the metabolic enzymes and they allosterically regulate the metabolism by increasing or decreasing the enzyme activity. So again, because these are experimental work, you can't work as you saw, just like the size of the metabolic pathway, we have thousands of reaction and so many protein enzyme interaction. So of course we need to validate. That's the thing which we do through experimental biology, but you can't work on so many reactions, and that's when systems biology comes into picture. So he showed that for 2, 3, or 4, maybe 5 maximum protein enzyme interaction. Then he applied the concept that if we can do it on a global scale, on a comprehensive scale, to get the idea how things look like. And that's where my job comes into picture. So first, how do I get the data? Uniprot, it's a database where you get information for all the proteins of multiple species. And each of the protein, if you just open a Uniprot page, it has an ID. It will have its function. If it will have information about its sequence. It will have information about its molecular function. It will have information about its biological process. All the protein details you get there. So I obtained we started the work on Escherichia coli K12 because it's the most studied lab organism. So computationally, how you get the data, I'm trying to explain that. Escherichia coli K12 proteome, that is all the proteins were obtained from Uniprot. Right now, we have around 4,500 proteins experimentally proven proteins in Escherichia coli K12. I got that list from Uniprot. Second, KEGG. KEGG, as I said, it gives you the list of all the metabolic pathways and the metabolic enzymes for different species. I got that list for Escherichia coli K12. So I have the list of all the proteins from Escherichia coli K12. I have the list of all the enzymes from the KEGG. Now what I did is basically I just extracted my metabolic enzymes and then found its protein partners. I'm working on protein-protein interaction. So now I have to find the protein binding partners of the metabolic enzymes. So I got the list of the pathways. For each of the pathway, I have the list of the enzymes. Now I have to find the binding partners of those enzymes. And during the rest of the talk, sometimes I will mention the binding partners as Interactor. So don't get confused. It's just the word play. So interactors are nothing. They are just the protein binding partners interacting with the metabolic enzymes, as you can see in the last one. So I obtained that from Intact. Intact is a protein-protein interaction databases. One good thing about Intact is every protein-protein interaction they mention in the database, they have a published reference. So each of the proteins, so basically the base data, the principal data which I'm using for my work, all the interaction have been published somewhere. So basically you get the data from the Intact. I have the list of the enzymes. I just found those protein enzyme interactions. I found the binding partners. Now I have a list of protein enzyme interaction obtained from Intact. And then focusing on binary interaction. As I said, during the protein-protein interaction, there are two types, complex as well as binary. Now, right now, the multi-protein complex is basically in a multi-protein complex. If one of the protein is a metabolic enzyme, right? Currently, it is basically a bit hard to determine that which protein or interactor is actually binding to the enzyme in the complex. It's a bit complicated. So that's why we wanted to make sure that we want to work only on those protein partners or interactors which are actually physically interacting with the metabolic enzymes. That's why we just focused on binary. We are keeping the complex aside, but definitely we have plans to look at that in future. So that's the thing. We get the data, and then so this was the first research paper we published 1.5 years ago, where we just worked on glycolysis in Escherichia coli. We have glycolysis pathway. For the glycolysis pathway, you have the glycolytic enzymes, metabolic enzymes. we explored the characteristics of the protein binding partners, which are binding to the glycolytic enzymes. And we got some results. I'm not going into the results of this paper because right now what I'm doing, we are scaling up the project. I will explain that in just a moment. And we are redoing those analysis again. So I will explain that here. So this was the first paper. We just published it for glycolysis. But as we just saw, like in the metabolic this picture, metabolic pathway, glycolysis is just this small line baby. So if you are trying to find some effect on the metabolism, just working glycolysis is just not enough. It was a pilot study, it was an initial study we published on glycolysis, but the idea is to scale up. Now go for much broader pathway. So the idea right now, we are working on carbohydrate metabolism, whose results I will show today. But probably by the end of the PhD, we have plans to do for multiple pathways and maybe the whole metabolic if we get good data. So, so right now we publish it for glycolysis. Now we are scaling the project to carbohydrate metabolism. The first thing, when I have this protein enzyme data and where the enzymes are all carbohydrate metabolic enzymes, I'm trying to find the characteristics of the interactor, biological characteristics of the interactor, which are binding to the carbohydrate metabolic enzymes, that how they look like, what are their functions. So, gene ontology is a database. You can see it was published in NAR. So that's a paper. So, without going into further details, gene ontology basically helps you to determine if a protein is metabolic in nature or it's a non-metabolic in nature. And if a protein is an enzyme or if it's a non-enzyme, if a protein is an enzyme, it will show the protein with an enzyme commission number. So all the enzymes have an EC number, but all the non-enzymes doesn't have. So that's the basic classification I did to find that among the interactors, among the binding partners of carbohydrate metabolic enzymes, how many are metabolic enzymes, how many are non-metabolic enzymes, how many are metabolic proteins. Metabolic proteins are basically metabolic non-enzymes and non-metabolic proteins. We found that, and it also makes sense, is like majority of the interactor, you can see, is 43.2%. Majority of the interactor are metabolic enzymes. So carbohydrate metabolic enzymes majorly interact again with metabolic enzymes which are governing some other metabolic reaction. Also 15%, it also interacts with 15% of metabolic proteins, 19% of non-metabolic proteins, and 4% of non-metabolic enzymes. The interesting thing is, as this paper, it talks about protein interaction. I mean, it makes sense. Like a protein, two proteins of similar function, they always have the tendency to interact more rather than proteins of dissimilar function. So that's the same thing. These are the carbohydrate metabolic enzymes. The binding partners are also again metabolic enzymes. So that's what we found. It's and it makes complete biological sense. But the good, interesting thing was 9% were uncharacterized interactor. Uncharacterized proteins are basically unknown proteins whose function are still not known. They have been experimentally proven as a protein. Something have been looked up, but their function, exact function, is still unknown. So 9% of the protein partners were uncharacterized interactors. So which shows that there is still something which we need to do, because it's basically called a black box research where some things are still unknown. And if we try to find the function of these uncharacterized interactors, it may help us to find some new it will help us to develop some novel ideas from there. So that's the basic first analysis what I did. The next, as I said, I'm working on carbohydrate metabolism. Carbohydrate metabolism is divided into all these smaller pathways on the y-axis: pyruvate, glycolysis, PCA cycle, butyrate, glycosylate, and then goes to inositol phosphate. On the x-axis, it's the number of protein-enzyme interactions for each of the pathway. The numbers which you see at the extreme right of the bar, that's the number of enzymes for each of the pathways. So basically, pyruvate metabolism has 31 metabolic enzymes, or you can also say they have 31 unique metabolic reactions, because each reaction is governed by one enzyme. glycolysis has 26, TCA has 19, and the numbers in the blue, that's the number of the protein partners, the binding partners for each of the pathway. So one thing you can observe here, like pyruvate metabolism, they have 118 interactor, but the number of protein enzyme interaction is actually around 125. It's happening because one metabolic enzyme, of course, it's interacting with multiple interactors, but there is also cases where one interactor is also interacting with multiple metabolic enzymes. So that's why the number of protein enzyme interaction and the number of interactor in blue, they kind of different for each of the pathway. Uh, I don't know any proper biological inference here about why glycolysis by looking at this figure. But what I want to say, I mean, if you just ask a biology student, glycolysis and TCA and butyrate and pyruvate metabolism, which is actually a major part of TCA cycle also, they have high protein enzyme interaction. They must have high amounts of regulation happening compared to this secondary metabolic pathways, ascorbate and aldehyde, inositol phosphate, which have low number of protein enzyme interactions. So that's again a basic result. Going further, the next thing what we wanted to show is the conservation. So we are working on protein enzyme interaction, and if a protein enzyme interaction is co-occurring in more species, that means it's important. So, and you can say that that protein enzyme interaction is actually much more conserved. And a protein enzyme interaction which is occurring in low number of species, it is low conserved. So using the Acknowledgement databases, first it works on the basis on the concept of ortholog genes. Ortholog genes are nothing but they are the genes which have been derived from a common ancestor, and after a speciation event, these genes are found in different species but with same function. So Acknowledgement database exactly works on that. It gives you a list of all the bacterial species where the orthologs are found, the number of ortholog members. So if you just put a protein name, a gene name, or a protein ID on the Acknowledgement, it will give you a list. For example, pyruvate kinase. Its gene name is PFKA. You put pyruvate kinase or you put PFKA, it will show you all the bacterial species where its orthologs are found. So I did that for my data set, that is protein enzyme interaction. I got the list for the proteins or the interactor, the number of I got the list of the bacterial species for all the interactors which are binding to the enzyme. I got the list of the bacterial species for all the enzymes. So now looking at just at the number of bacterial species for the interactor or the enzyme differently doesn't make sense. We are working on interaction. So the most easy thing what I did, I have the list of the bacterial species, list of the ortholog members for the protein. I have the list of the bacterial species, ortholog members for the enzyme. I just found the overlapping species, which shows that how much protein enzyme interaction is actually conserved. I found those overlapping number, and that's the number which I plot in the y-axis, which basically shows how much that protein enzyme interaction. So if that protein and enzyme, if they're interacting in Escherichia coli K12, and if that protein and enzyme is also again found in another species, that means they are interacting. So that number was plotted in the y-axis. So that's this is a box and whisker plot. Those it's not a bar, it's a box and whisker. So the point where this line starts, it's basically 0%. This first shows 25%. This is the median, 50%, that's 75%, and the top where the line ends is the 100%. So that's how a box and whisker plot, and it's following just the same concept. This line which goes, it's the average of all the values, which is 2779.5. And for each of the plots, you can see a big black dot, which is the average for each of the pathway. Now, first of all, as I said, these brown dots, these are protein enzyme interaction, which are showing their number of ortholog members or number of bacterial species for and each of the dot is showing that. Each of the dot is a protein enzyme interaction. So the dot at the top, so it is basically GAP A, which is a metabolic enzyme of glycolysis, interacting with gap B, which is the interactor. It's a high conserved protein-enzyme interaction because this interaction is found in more than 6,000 bacterial species. Similarly, another one, RPE, which is an enzyme, interacting with DNA, N, which is the interactor. It is also found in more than 6,000 species, part of pentose phosphate pathway. The enzyme is a part of the pentose phosphate pathway. So these are high conserved protein enzyme interaction. The one which comes in the dots which are at the bottom, they are the low conserved because they are found in less than maybe 10 species. So again, making a reference, making a biological inference from here, if you just follow this average as a threshold for like making it like anything coming above that is highly conserved and anything coming below that is low conserved. I would say like the dark green TCA cycle, glycolysis, pentose phosphate pathway, as you can see, the average, the black dots are coming above this line. So basically they are more conserved compared to starch and sucrose metabolism, amino sugar and nucleotide metabolism, ascorbate and aldehyde metabolism. So again, the secondary metabolic pathways, the protein enzyme interaction of these pathways are low conserved. The protein enzyme interaction of TCA cycle, glycolysis, pentose phosphate pathway, and interestingly is we need to find out why it's happening, but glycosylate and dicarboxylate metabolism is also showing high conservation. So basically we did that, so sometimes protein enzyme interaction are highly conserved, sometimes they are low conserved depending upon the metabolic pathway. The next thing what we did, we are still in the middle of this analysis. I will not go much into details, but as I said, this work depends a lot on the data. So this paper, the quantitative and condition dependent Escherichia coli proteome by Alexander Schmidt, he basically does experimental studies where he tries to measure protein concentration, protein copies per cell. So basically we wanted to assess that among the enzymes and the binding partners, how much the abundance is different. So how much enzyme is abundant compared to their interactor. So in the previous plot, where you just saw one box plot, for this we are not showing as an interaction, but we are showing the two proteins separately. So for each of the pathways, amino sugar, ascorbate, for each of the pathways you will see two box plots. The one on the right, a bit darker, they are the enzymes, metabolic enzymes. The one on the left, they are the interactors. So again, the black dots for each of the box plot, they are the average. So, and this is, coming back to the paper, quantitative and condition dependent Escherichia coli proteome. Alexander Schmidt, we have, as I said, around 4,000 proteins. He did the experiment for 2,600, maybe some experimental, sometimes just can't, basically he didn't do the for 4,000 proteins, there can be many experimental reasons. So there were 2,600 proteins what he used in that data set, and he basically found the protein copies for all those Escherichia coli for Escherichia coli in 22 different conditions, like starting with LB, glucose, chemostat, stationary. There were 22 different conditions, and we are using that data to do our analysis. And this is just one plot, LB. We need to do the same thing for other conditions also. So right now, and basically the paper which we are planning to write, we will probably not show all the 22, it will be a big mess. But we actually need to get this plot for all the 22 different conditions, because metabolism is something which is very much condition dependent. And we want to see that which pathways or which pathways, and by pathway I mean the enzymes and interactor, if they are showing any fluctuations in particular condition or not. This is just LB. Looking at the LB, as you can see, for the amino sugar and nucleotide sugar metabolism pathway, enzymes are low in abundance compared to interactor. As you can see, the black dot, the big black dot. For TCA cycle, for glycolysis, the enzymes are higher in abundance compared to the interactor.
So when you you're saying just get TCA cycle enzyme and the interactor.
Yeah.
But at formula flux.
Yeah.
Okay, all enzymes are not equally important, right?
Yeah.
There could be rate limiting enzyme. So why are you're talking about the same breath, the TCA cycle enzyme? Would it be different for a rate limiting enzyme or for an enzyme which other than rate limiting enzyme?
Yeah, I mean the thing is
Will we miss something if you don't look at for each enzyme and its interest?
actually for the Escherichia for the TCA cycle enzymes, yeah, there have been some cases when I'm talking about the TCA cycle enzymes. If they have 10 enzymes, there can be some 2 or 3 enzymes which are not interacting at all. There were just 7 enzymes.
Yes.
Yeah. So yeah, I mean in Escherichia coli, if you talk about the metabolic, exactly. If we have 1,000 enzymes in Escherichia coli, 52% of the enzymes are actually not forming an interaction. We are just focusing on 48%.
So then if you're using all the name as TCA cycle enzymes,
yeah,
but looking at interactors, will we miss some regulatory aspect of that?
I mean, that's the thing. So the rate limiting enzymes, what we are trying to find, we tried to find a correlation with the number of the binding partners they have, but we didn't find any significant result there. And another challenge is, as you said, the rate limiting enzyme. It's very tough to get the enzyme activity values, the turnover number for all the enzymes. We don't have so much experiment. So another plan was what we are doing. If I get that turnover number, that value, I can also correlate that with the number of partners. So if they have high activity, does that mean they have high partners? But again, that the lack of data. So that's the thing. Yeah. So, but I'm just saying because the enzymes are very much higher, they compared to Interactor, they are high in abundance. But that's not the case for the other pathway, like for amino sugar, for ascorbate and deltarate, the enzymes are low in abundance compared to Interactor. And for the pentose phosphate pathway, they are kind of at the similar level. But as I said, this is just LB. I need to do the same thing for all the others, and then I will try to make some inference if there is a vast difference or not. So I'm right now in the middle of that. So on the left, this is just the figure of the big metabolic pathway picture from the cake which you just saw. So we are covering now this much. This is the carbohydrate metabolism, and I'm highlighting all the pathways. The numbers in the bracket, they are basically showing the number of interactors per pathway, average number of interactors per pathway. This is just like a summary figure which we are going to use. And again, why are we doing this work? Coming back to that, the ultimate idea is we are doing conservation, we are doing abundance, and all this analysis is to find those, as I said, 3, 4, or 5 candidate protein enzyme interaction, which will be again sent to an experimental biology lab, and they will validate it. This is the table which is from the last paper. So I just put it here, and we are again planning to get some, just for glycolysis. We are again planning to get some candidate protein enzyme interaction looking at carbohydrate metabolism, because ultimately validation is extremely important. So that's the thing. So biological research is going towards the area where if you have so many data, thousands of reactions, thousands of protein enzyme interactions, you can't randomly pick experimentally that we have to target that. So rather than that, people like us, systems biology data analysts, we analyze the data using some biological knowledge. We make some predictions. So rather than just randomly picking up or rather than wasting time there, we predict some candidate protein enzyme interaction and work and do the experiment on those 5 or those 6 protein enzyme interaction, because the chances of being successful on these predictions will always be higher, because these predictions have been made again through a biological knowledge. So that's the manuscript process we are right now working on. Network of protein enzyme interaction regulate carbohydrate metabolism in Escherichia coli and other bacteria. Other bacteria is because of the conservation analysis which we just saw. So that's the idea. Future studies, there are some things. First, as I said, we are just focusing on binary interaction. We are making sure that the protein or the interactor is actually physically interacting with the enzyme. But as I said, there is a lack of understanding in a multi-protein complex. If one of the protein is an enzyme, which one is actually binding to the enzyme? That's kind of difficult to find. But definitely I have to find something there and look at the protein enzyme interaction making multi-protein complexes, because again there are a lot of papers where complexes play a huge role in metabolic adaptation. So that's the thing which we are right now not working on, but that is of course one of the study. Perform this investigation for other species like yeast and humans. We are just doing for E. coli. Maybe do the same thing for yeast and humans and get a comparative study that how things are different for these three species. Scale up the analysis for other pathways. I'm headed for glycolysis. Right now we are going for carbohydrate metabolism. And then by the end of the PhD probably some more pathways, some whole metabolic. As I said, the data basically this data, so most of the studies when the people do this protein abundance studies, most of them they do is in the carbon sources. Like the condition, as I said, that is LV is fine, but we have glucose, glycerol, chemostat, osmotic stress glucose. So these all were very much carbon related condition. So that's why we again thought that maybe get something around carbohydrate metabolism. But if we have a data where we can test the protein abundance for other pathways like lipid metabolism or amino acid metabolism. So if we get some cell environmental conditions like that, maybe we can of course do that for other pathways for metabolism, because metabolism is ultimately connected. So till you are not doing complete metabolism in order to get published in a very good journal, I guess that's the thing. But carbohydrate metabolism, it's the compared to lipid, amino acid, uh, energy, the lot of broad pathways, carbohydrate metabolism is the one which have maximum number of enzymes and maximum number of protein enzyme interaction. And many diseases are related to carbohydrate metabolism. So it's basically it's worth to work on carbohydrate metabolism, but maybe in future do it for both. And of course, as Bhushan just said, the flux metabolic modeling analysis, we are trying to find the effect on metabolism. So there is some computational analysis which where people do just the metabolic modeling, where people try to model the metabolism in a species uh, based on. So basically, there is software. You give all the genes, you give all the enzymes, and you give all the metabolic reaction, and you give a particular growth rate, a constraint, and then you simulate, and it gives you basically some metabolic modeling, like in that growth rate, how the cell is performing. So that's the metabolic modeling, and that's basically if we find the candidate protein enzyme interaction for those reaction, how the rate is changing, the flux is changing. That's the area which you need to do. I'm actually not doing that right now, but in the first year of my PhD, I've written some reviews around that, so I know how it is done. But of course, when you start doing that, it's actually a bit challenging. It's a new thing for me. But of course, for this project, that's also the thing which you should do. And last, the thing is, why are we doing what are we doing? As I said, it's just for Escherichia coli K12, but maybe when we scale up to other species, do it for other species, finding candidate protein-protein interaction or protein enzyme interaction for globally known metabolic disorders like obesity and diabetes. These are very much far-reaching applications. I completely agree, but these are ultimately metabolic disorders. So if during obesity or during diabetes, if you are able to predict or find those candidate protein enzyme interaction where metabolism gets regulated hugely, they can be used as drug target. Similarly, knocking out Escherichia coli O157, it's a pathogenic E. coli causing foodborne diseases. It is able to do that because it is surviving. It is surviving because its metabolism is active. If you again find those protein enzyme interaction where metabolism gets affected, you can again use that as drug target. Last is developing new metabolic engineering strategies for efficient biofuel production. Escherichia coli is regularly used for biofuel production. So developing new strategies by basically, if we have the idea of those candidate protein enzyme interaction, we can develop new strategies. Last, acknowledgement. So this is Dr. Peter Woods. This is my primary PI. Dr. Stephen Fong, co-PI. He is the protein-protein interaction guy, and he is the metabolism guy. And as I said during the initial stage of my talk, I'm exactly working on the middle. So that's why this collaborative project was started. So he's a protein guy, he's a metabolism guy, and I'm basically, they both are my PI, so I'm working both in the them. And as I said, you need to have some collaboration where people will do those experiments. So Dr. Milton Sayer, he's in UC San Diego, he's a collaborator. So whatever predictions I will make, and we have made, we will, he has students who will go there. He has students in his lab where we will send our data and they will do those experiments to validate the things which I'm just showing. This is Richmond City. I just wanted to put a picture. It's around James River. It's the capital city of Virginia, and it's a nice city. The city sleeps very fast, and and I like to say to everyone, it may sound weird, but like I like to say that it's exactly like the Baroda of USA. It's a small city, but you get everything to live a comfortable life. It's just like that. Lastly, I would like to thank Dr. Pushpa Robin for giving me the opportunity to give the talk here. Dr. Sarita Gupta, as I said, I was part of the first batch. She was the director of the program, and under whom, basically under her guidance, we did all the training. So I will always be thankful to her. Of course, the CMB faculty members who are here, I would like to thank you all. And last but not the least, all the students of the cell and molecular biology, thank you for attending the talk. And that's all from my side. And if you have any questions, thank you.
I know that now the systems biology is very useful for predicting and also your computational analysis before you directly go into the wet experiment and do it. Right? What are the percentage of error that when you have predicted that this protein or enzyme interaction will be there? What is the percentage of it? Not when you are verified with the wet experiment.
Yeah.
What are the chances it will not be there? I mean, that
Absolutely.
Does it happen?
Yeah, there have been some. I'm not saying so. If I send 5 candidate protein enzyme interaction, I should say the percentage of error should be 10% because among those 5, experimentally at least 2 or 3 should be proven. But I will not deny the fact that all the 5 will be rejected. But if it gets rejected, again, I will definitely there was some flaw in my modeling approach. I have to do that again analysis and predict some new. So.
And for computation, I mean, when you are doing the computational analysis,
Yeah.
do they accept without verifying with the web experiment or?
Yeah, they do actually. Right now, it's a bit honestly. I mean, Nature Chemical Biology. They have. If you just go to the journal scope, they are accepting papers without experimental. So they just work. So things are changing. I mean, that's the thing. But definitely, but I can't publish in Nature Biotechnology. There, they will want experimental biology. Same. And so definitely, in order to publish in a good journal like a top, if you have the experimental backup, that's always better.
So in your lab, there is no wet lab. Means biopsy or yeast two-hybrid system or any other technique for the protein prediction.
No, I'm not doing, but we have the collaborator where I will send the predictions.
Have you mentioned the analysis, modeling of the metabolism and the human microbiome? Right?
Yeah, that's it.
So, does it involve any machine learning techniques?
So that paper was it was just a review. So that was completely based on metabolic modeling. So what I did is like that review. I'm talking about the microbiome, which have all the bacterial species. I'm not talking about. I don't think I remember mentioning machine learning, but we just mentioned all the studies where people have done metabolic modeling for microbiome. So that's
But it varies due to the geographical reasons. Right?
absolutely.
That is very problematic.
Absolutely.
Microbiome profile doesn't have a parallel system
Yeah.
in most of the people.
Yeah, and we just focused on 3 or 4 microbiome, like the oral one, then the gut microbiome, vaginal microbiome, these 3-4 things, and then we just published a review.
So most of the regulation happens at the multi-protein part,
Yeah,
right? And then you are focusing on binary.
yeah,
So how the very
I mean,
important part is
yeah. I mean, I should not say that every time regulation is happening at the multi-protein. It was like the study. The idea which we are trying to find is basically proteins which are actually physically interacting, as I said. So, and as I showed you 4 or 5 studies, these are all binary interaction. The studies which made us start with those projects, they were all binary interaction. They were not multi-protein complexes. But I agree that we have to look at the complexes also to get to complete the full picture. Yeah.
How about transient and weak interactions?
Yeah.
They are also covered and
All the binary, I guess, all the binary interaction are transient, and the one which are multi-protein, they are the stable ones. The transient are the one they just interact, they do their job, and they just leave. So that's the thing.
One more last question.
Yeah, go ahead.
Since you're
Students can ask.
Last question.
okay, that's-
So, yeah.
since you are from, it's a non-academic question. So
Yeah.
since you're from the very first batch, so
Yeah.
you know how this department got established and all.
Yeah.
So can you give some tips and tricks to our students?
Yeah, I can. In what area do you want? I mean, I can. Yeah, like tips and tricks for what? I mean,
Means
for the
since they're from post-COVID, so from COVID era, so you can get to know that. So you can give some means around how to cope up with the subjects and all.
cope up with the subjects. That's my again. I will ultimately I will again go into academics. Basically, if people are who are wanting to PhD after CMB, I just want to say the net. Basically, if you want to do in PhD in India, you have to clear net at some point of time. I just want to say the syllabus which you have for all the 5 years is basically the net syllabus. So if you the thing which we also didn't used to do basically, if you want to clear net, once the semester is over, we used to forget our subject. So you always need to revise. I'm sorry, I don't know, I'm not getting the question, but the thing is like, yeah, if anybody wants to do PhD in India, you have to clear net, and the syllabus is exactly, and you just have to revise. Biology is a subject which you forget very fast, and some of the basics which I just talked today, I again revise, which are the basics. Again, I have to read and basically, oh yeah, so that's the thing. And abroad application, I guess, as Kushwaram said, I can help with them if they have any questions. So, and yeah.
I have a question. Like one of the most basic differences that we know in prokaryotes and eukaryotes is that in eukaryotes we have post-translation modifications.
Yeah.
Yeah. So for instance, if in your future studies, like you said, we'll be comparing the eukaryotic models as well. So for instance, in E. coli, you are getting two proteins which are interacting, but the same proteins are interacting in eukaryotes model as well.
Yeah.
But one of the proteins which is interacting, it might have undergone post-translation modification, for instance, phosphorylation. So how will you like differentiate? What will be the strategy to make sure the interaction or the effect of the protein-protein interaction that is taking place is because of just the interaction, or maybe because of the post-translation modification that is happening?
I mean, I will actually not focus. So basically, if we have if you have a protein in the E. coli, it's doing a metabolic reaction like glycolysis. Basically, it's happening in E. coli as well as in humans. I will probably I will think about that looking at the post-translation modification. But as in right now, if I have the enzymes which are basically part of the human, I will put that in the database intact, and I will get the protein partners. And as I said, those all interactions which they are showing there, these are proper protein-protein interaction study. So again, the base data which I'm going to use, that will always be a protein-protein interaction data, which are coming from a paper. So, but how to tackle post-translational modification, I will look into that. But to answer your question, the data which I'm basically using, it will always be either through yeast hybrid, either through affinity purification, the general protein-protein interaction method. Yeah.
not the bio-ID or something, you are not.
Biobrain.
Proximity ligation, you are not taking.
No.
No.
What is it?
Proximity ligation,
Yeah.
bio-ID or Apex-based interactions, you are not taking into the consideration. So you are missing a lot of transient and weak interactions.
Any other questions from the students?
Any other question?
If you have any questions about
General questions.
general questions, yeah, about the application process or anything, so I can help you.
You can always take his email ID. But they can,
Yeah, definitely. Yeah,
you can leave and then they can talk to you.
Yeah, that will remove all the information.
Thank you so much.
Very nice.
Yeah, all the best.